Hello everyone, my name is Erica Almance and my partner Suprina Shrestha and I are from the UTA Department of Earth and Environmental Sciences. Our supervising professor is Dr. Ricardo Sanchez Murillo. We will be talking about the use of water isotopologues to reveal versatile plant water sourcing strategies in a highly altered urban landscape.
Our study site, the Fort Worth Botanic Gardens, is an example of urban green infrastructure, which is any system that serves to mitigate water pollution and floods from flashy stormwater episodes. However, there is little understanding of the role that plants play in partitioning the urban water cycle. So, what water sources do different species prefer?
We used naturally occurring tracers, or water isotopes, to examine how precipitation inputs are stored in the soil and taken up by plants. Each water sample, the soil, stems, roots, and precipitation, has its own value of delta O18, the ratio of heavy oxygen to light oxygen in the water. We call this isotope value the isotopic signature. We can compare the signatures of the plants to the signatures of the precipitation and soils at different depths. Signatures that match those of the plants are the sources of water that those plants are using. The three tree species that we studied were box elder maple, cherry laurel, and elderberry, which are native and common species in the DFW area.
We took samples of the xylem wood from stems and roots, as well as soil samples at different depths, and centrifuged those to extract the water. We then compared the water from stems and roots to the extracted water from soils, alongside samples of throughfall, which is the precipitation that falls through the forest canopy and ends up on the ground.
We conducted a gravimetric analysis of the plant material and the soils and found that there was no significant relationship between the amount of water extracted and the isotope values. In other words, the amount of water extracted does not cause a dilution effect.
Our isotopic results show that box elder maple was the most enriched or high in oxygen 18, suggesting a longer water residence time or a preference for distant water sources. Overall, cherry laurel and elderberry exhibited isotope values that agreed with recent throughfall events and shallow soil water sources, suggesting quicker water uptake. STEM delta O18 values showed a strong temporal trend from high variability in winter, fall, and summer, with more uniform values in the spring. Roots had increasingly enriched delta O18 values from winter to summer.
We used a Bayesian mixing model to determine the contribution of water sources, through fault and soils, to the mixture, or the stem and roots. Here we're presenting box elder maple as an example. During the active growing season of spring, box elder maple's primary water source came from soil depths of 0 to 12.7 centimeters, or 5 inches. During the summer, the water-stressed period, box elder maple preferred shallow soil moisture from the few sporadic rainfall events that occurred.
Our results contribute to the current water extraction method debate for stable isotope analysis, as well as the understanding of plant water uptake strategies of these three representative plant species in an urban green landscape.
Our future research includes sampling more individuals of the same tree species, sap flow measurements, and an analysis of the diurnal variability of stem and soil isotope ratios.
Thank you to the Fort Worth Botanic Gardens, UT System STARS program, and the UTA Office of the Provost. Thank you for watching.